All right, students. So today we're going to do 2.3b. So that's the second part of our lab. So we're going to go over here, 2.3b. And so we can look at these questions here. So what we're going to do first is we're going to use this procedure, and we're going to use the procedure from the other day, and we're actually going to draw on on the paper that we have here, and we're going to label each of these with the bacteria that are on each part. So what we're going to do is that you're going to use, so you would use colored pencils in class. So a red color pencil and a blue color pencil. The red color pencil is going to be for strain 1 bacteria, and the blue color pencil is going to be for strain 2 bacteria. So we have red. Red for strain 1 and blue for strain 2. You're going to color in on your paper. I'm going to do mine on the computer so that you can see it. But let's go ahead and check it out. That's not what I wanted to show, but let's go here. So it says we're gonna look back on this and we're gonna see where did we put each of the bacteria. So if we go back to 2.3a, let's look at the actual plates. So on the left side of each plate, on side 1, we added strain 1 bacteria. And then on the second side, on side 2, we put strain 2 bacteria. So that means that on each one of these plates here on the left side, I'm going to put on the left side, I'm going to put strain 1 bacteria, and on the right side, I'm going to put strain 2 bacteria. So on the left side, I'm going to make that red, and on the left side, I'm going to make it red. On the right side, I'm going to make it blue. So let's see if I can find a good one that fits here. Well, let's see. So we are looking at a semicircle. So I'm trying to see if maybe this one might work. Let's see if we do this. There we go. Okay, so I want it to be red on the left and then blue on the right. So, and again, this is obviously going to be different for you because you're using a color pencil, and honestly, a color pencil is probably a better idea. But it's going to be red on the left side because it is. That's where right. So red here on the left side because this is where strain 1 is, and it's going to be blue on the right side because that's where strain 2 is. And this is going to be the case for every single one of our plates. And yours hopefully will look a little bit better than mine. But we're going to put red on the left side and blue on the right side for each one of these plates. Right? Because on every single plate, we put strain 1 on the left side and strain 2 on the right side for each of these. Cool. So here it says, why are some bacteria not affected by antibiotics? So in the antibiotic resistance lab, part 1, we used 2 types of bacteria, strain 1 and strain 2. Each type of bacteria had resistance to one type of antibiotic. Typically, antibiotics stop bacterial cell growth and kill the bacteria. However, some bacteria develop mutations, changes, in their DNA. Mutated bacteria will not be affected by the antibiotics. They have antibiotic resistance. So why are some bacteria not affected by antibiotics? Well, because some bacteria develop mutations in their DNA that makes them not affected to antibiotics. So we call these antibiotic resistant bacteria. So this would be so some bacteria develop mutations. Those mutations make it resistant. So mutations are just changes to our DNA that make us more likely to then change the qualities that we have. Number 2, why are antibiotic resistance bacteria harder to treat? Well, it says bacteria with muted DNA reproduce and pass resistance to the next generation, resulting in an antibiotic resistance population. These bacteria cause infections that are harder to treat because they resist the drugs and chemicals that once kill them. In this lab, strain 1 bacteria develop resistance to the antibiotic streptomycin, while strain 2 bacteria develop resistance to the antibiotic ampicillin. If the bacteria resists a specific antibiotic, it will continue to grow and reproduce. So why are these harder to treat? So these bacteria cause infections and are harder to treat because they resist the drugs and chemicals that once treated them. So if we're looking here at strain 1, so remember strain 1 bacteria. Strain 1 bacteria was the one that we put on the left side. Strain 1 bacteria, we know that this strain 1 bacteria has developed the resistance to the antibiotic streptomycin. So when I come down here, my streptomycin is what strain 1 is resistant to. Strain 2 is resisting ampicillin. So something has resistance. That means that if I put streptomycin on the first strain, nothing is going to happen to it. When I look at it when I look at the bacteria, if I put the strain 1 on streptomycin, I should see that the bacteria still grows because it's resistant. It doesn't matter that that antibiotic is there. It can grow regardless. Strain 2, however, has ampicillin. So, or strain 2 rather is resistant to ampicillin. So that means that if I put strain 2 on the ampicillin, nothing will happen to it. Strain 2 will still be able to grow even though ampicillin is there. So right now we're going to make a prediction. We're going to make a prediction of what we think we're going to see after some time. So this first plate, what antibiotics are in this plate? So on this first plate, this is just LB. There is no antibiotic on this plate. So I'm going to circle none. You can't circle because I can't circle on this side, but I'm going to just bold none. The second one has this STR is for streptomycin. So the second one has the antibiotic streptomycin. The third one has the antibiotic ampicillin. And the last one has both antibiotics, streptomycin and ampicillin, has both of them. So what are the strains that can grow on this plate? So on this first one, there's no antibiotic. So that means that both strains should be able to grow. There should be no problem for either of them to grow. There's no antibiotic there to prevent them from growing, so they should be fine. So I should see growth of both strain 1 and strain 2. So I'm going to make strain 1 again the red and strain 2 the blue. And so I'm going to put that the strains that are going to grow are going to be both strain 1 and strain 2. There's no antibiotic there. They should be fine. On the second plate, right, on the second plate, we have the antibiotic streptomycin. Strain 1 is resistant to streptomycin. So that means that it should be able to grow even though there's that antibiotic there because this strain 1 is already resistant to it. It already knows how to combat it. So here I should only have the left side. Only the left side should grow. The right side will not grow. And the reason it won't grow is because this second strain is resistant to ampicillin, not streptomycin. So this means that it can only resist the antibiotic ampicillin. So on this side, when we're adding ampicillin, the only one that's going to grow is the one that has resistance to ampicillin, which is strain 2. So it's going to be the opposite of this one. Here we should see growth of the second strain and not the first one. And the reason for that again is the second strain is resistant to ampicillin, which means that it can grow regardless of whether or not there's ampicillin present. So that means that on this plate, we're only going to see strain 1. We should only see strain 1. And on this plate, we should only see strain 2. Then on this plate, we have 2 antibiotics present. So we have both streptomycin and ampicillin. So the question is, which of these will grow? So let's see, strain 1 is resistant to streptomycin and strain 2 is resistant to ampicillin. However, strain 1 is not resistant to ampicillin. So that means if there's ampicillin present, strain 1 cannot grow. And this plate does have ampicillin. It has ampicillin and streptomycin. It has both. So I should not see growth on the one side because there's ampicillin here that will kill strain 1. Because this strain, again, is only resistant to streptomycin. It is not resistant to ampicillin. Strain 2, on the other hand, is resistant to ampicillin. It is not resistant to streptomycin. Since this plate also has streptomycin on it, then I should probably predict that the second one will also not grow. So basically, we shouldn't see any growth on either of those plates if we are correct. So what we're going to do is we're going to actually now look at our results. We're going to see if we were right with our predictions. So we're going to grab our bacteria from the incubator, and we will check them out. So I'm going to go grab the bacteria from the incubator and see what we got. So moving over to the incubator, we're going to look at our samples and see what we got, see if we got anything interesting. So we should have a few things. We should have our plates, and we should have the 4 plates that we set on the other day. And we should also have our mating plate. And it looks like we are right on the dot. So I'm pulling these out of the incubator, pulling them out here. So I have my mating plate, and then I have my 4 plates with the different kinds of antibiotics on them. So I'm going to come and bring these over here to my table so I can work and look at them. So let's see if our predictions were correct. We have to analyze our results now and see if we were correct in what we were looking at. So let's see. Let's look through our procedure to make sure that we're following the correct instructions. So we got the 5 dishes from the incubator. We're going to turn all those petri dishes over to the bottom part, and then we're going to look for the growth. And we're going to see where the bacteria has grown and has not grown. And we're going to see if these predictions that we had here were correct. So let's turn these petri dishes on the bottom, and I'll put them here so that you can see it. So we have our petri dishes. We have we have LB, we have LB plus STR, we have LB plus AMP, and then we have LB plus STR plus AMP. So we have 4 different. So, and then we have our mating plate, but we're going to keep this on the side for now. So let's look at our plates. Let's look at what we actually have. So what we're going to do is if we're going to check the growth. So if we see growth, we're going to put a plus. If we see no growth, we're going to put a minus. So on this LB plate for strain 1, so let's see. So what I'm seeing here is that I have growth on side 1 and I have growth on side 2. So I have growth on both sides, which means that I'm going to put a plus for strain 1 and a plus for strain 2. I see growth for both of these. So let's go back over here. I see a plus, so plus here and plus here, growth for both of these. The second one, let's look at the plate with LB and STR. For LB and STR, I see growth on the side that has 1. I see no growth on the side that says 2. So that means that only strain 1 of the bacteria was able to grow. The second strain was not able to grow. So I'm going to put a plus here and a minus here. Okay. Then let's look at the plate with LB and ampicillin. So LB and ampicillin, I have no growth on side 1, but I do have growth on side 2. So I'm going to put a minus for 1 and a plus for 2. So strain 1 was not able to grow, but strain 2 was able to grow. So a minus here and a plus here. Then for the last one, LB plus STR plus AMP, I'm going to see, if I look closely at this one, what I see is that there's no growth on either side. There's no bacteria that grew for either of these parts. So when I come back here, I'm going to put a minus because strain 1 and strain 2, neither of them had any growth. So I'm going to put a minus for both of those. And there we go. Let's look back over here. So I'm going to take a picture of my Petri dishes with my phone. I'm going to use my phone to take a picture. Usually I would not recommend that you use your phone while you have gloves on because you want to make sure that you don't contaminate anything. If you have bacteria on your hand and then you use your phone, now you have bacteria on your phone. But I am going to take that risk just for you all. Okay, so we took a picture. So now what we're going to do is we're going to get 3 new Petri dishes, one that says LB-STR, one that says LB-AMP, and then one that has LB-STR-AMP. So we want to have 3 Petri dishes. So let me go grab my Petri dishes. So I got my plates. So I got LB plus AMP. I have my LB plus STR, and I have my LB plus STR plus AMP. So I have my 3 plates, which is exactly what I want. So let me cross these guys all off. I'm going to take off the lid of all of these. Take the lid off of each of these plates and have them just prepared here because I'm going to use them in a bit. So I'm going to put these all on top, and I'm going to take the lid off of each of these to make sure that we can then streak our plates. So then if you remember, we have a little plate that's called the mating plate, right? This is where we put the 2 bacteria together. And what we're going to do is we're going to put these bacteria on each one of these plates, right? These 2, strain 1 and strain 2 have gotten some time now to hang out, to be friends. And now we're going to put this bacteria on each one of these plates to see what happens now that they've mixed together. So coming back here, so we remove the lid from the 3 Petri dishes. We're going to grab this mating plate and we're going to remove this lid. And we're going to take bacteria and we're going to streak the plate that says LB plus STR. So I'm going to take the top of this. I'm going to grab a loop and I'm going to put it on this plate here, the one that says LB plus STR. I'm going to take bacteria from here and I'm going to streak onto, which is just another fancy word for spread. I'm going to take some bacteria from here. I'm going to streak right on top of this plate all over the whole surface. Make sure that there's plenty of bacteria all over the place for these bacteria to grow and to see what happens to them. So let's make sure that's nice and mixed. Mix it up really nice. Okay. I'm going to put this, right, every time we use one of these, we're going to put it in the waste. I'm going to put this in the waste. Let's look back here, make sure we're following our procedure. All right, so we streak the plate, we put the loop into the waste container. So now we're going to grab a new loop and we're going to now put these on the plate that says LB plus AMP. So this plate, the LB plus STR, this plate is done. We're going to grab the plate that says LB plus AMP. I'm going to grab a new loop and I'm going to take bacteria from the mating plate, from my mating plate, and put it on. So right, I'll grab some bacteria, streak over the surface, and I'm going to put it on the STR or the LB plus AMP plate rather, sorry. So I'm going to streak right on top again, just very gently spreading over the surface. Again, the agar is very fragile. So you should just barely be scraping over the surface. Want to make sure that it's not, right? You don't want to rip the agar. The agar can rip very easily, but just make sure that you see that you have put the bacteria all over the surface. So then again, always putting our tip here, or always putting our loop in the waste container. All right, returning back to our procedure. So we did this part here and this part here. We're going to grab a new loop, and now we're going to put these bacteria on the LB plus STR plus AMP plate. So that's the one that has all three of the all three parts: the food, the streptomycin, and the ampicillin antibiotics. So let's grab a new loop, and we're going to streak from our mating plate onto our LB plus STR plus AMP plate. So I'm going to streak all over the surface again, very gently. You want to make sure you don't want to rip your agar. You want to make sure that it grows nice and evenly. So we spread all over the surface. Okay. And then put this loop into our rice container. Excellent. So here we go. We're going to put the lid on all three of the plates. We're going to put this lid on, and then we're going to put, just as we did before, our the class and our initials. So I'm going to put the lid on all of these plates. I'm going to put my class, and again, for you, it would be class A, B, C, or D. For me, I'm going to put class Z and AC. Same here, class Z. Then we have AC, my initials. Same here. We're going to do LB, or we're going to do class Z, and then AC, my initials. And then those are those three. So we put our class and the initials on all three of these plates. We're going to turn these Petri dishes over to the bottom part, and we're going to tape them together and then bring them over to the incubator. So let's put these all on their face down side. You want the top to be on the bottom. We'll flip them over to like this. This is how bacteria like to go inside the incubator. They're kind of funny that way. And then we will put these, we're going to tape them together using this tape, so that then when they go in the incubator, they can stick together and they're not kind of moving all over the place. But here we have our three plates, and they are now taped together for us. Okay. So we're going to take these guys over to their, so also you want to cover this mating plate. Here, make sure it's covered. We don't really need this anymore, but we're going to take these over to the incubator so that it can grow overnight. So let's take these over. Again, the incubator is just where we put bacteria for them to grow. It is nice and warm on the inside, and it keeps them in the conditions that are ideal for their growth. So we'll take these, put them right inside the incubator face down, and we'll leave them there for a day for us to check. And again, I'll just make sure that the temperature is set to 37 degrees, which is the temperature we want them to be at. So let's come back over here and let's check out what we've got going on. Make sure that we've gone through our whole procedure. And yes, so we turned them over to the bottom part. We put tape. We brought the three dishes to the incubator, and we put the lid on the, let's put the lid, we put the lid back on the mating plate, right? Just put the lid back on, make sure that it's closed up. And then, so we're going to return all these plates to your teacher. You're going to bring them to me, and then you're going to use a sanitary wipe to clean your table. But that takes us to the end of 2.3b, and I will see you all on the next piece.